Great. Okay, so thank you for inviting me to speak in this event. What I'd like to tell you today is how we were able to identify COVID-19 responsive T cells from longitudinal data at even for even one specific patient. And this is work that was done in collaboration with our colleagues in Moscow and was really led by Asia Minervina and Misha Pogorelli, in collaboration also with people in Paris, Thierry Morin and Miriam Bensuda Karaichi, and collaborators in Kiev. So it's a joint effort of the community. And so let me start. So our data consists of 2 donors that basically a couple of days into quarantine developed relatively mild symptoms of COVID, and then antibody testing verified that they were indeed COVID-19 positive. And after quarantine ended, the 2 donors donated blood at day 15, 30, 37, 45, and 85 post-infection. At each time point, there were two replicates made, and the blood was sorted into CD4 and CT8 pools. And at certain time points, we also got memory data. So then the RNA was sorted out, and TCR alpha and beta repertoires were sequenced independently independently. Additionally, for these 2 donors, we have 2 time points pre-infection from 2018 and 2019. So we'll use that later. But the first thing that we did is we just looked at the longitudinal clonal trajectories, and we did a PCA analysis of the full trajectories, and we identified 3 clusters. And the data looks very similar for the 2 donors. There's a cluster that is basically where clones have stable frequency over time, so nothing much happens. That's the cluster you see here. Then there's a cluster, the green cluster, which from day 15 to basically clones that contract from day 15 in frequency to later days. And it's a stable contraction for the 2 donors. And this is basically what was expected, because the peak of the infection was expected to be before day 15 or thereabouts. So that's sort of what we thought we'd see. And then there was this second cluster, which is a little bit more mysterious, that the clones expanded from day 15 to day 37 with a peak at day 37, and then they contracted again. So this seems to be some sort of later activation. And this, as I said, this was a little bit less expected at that point. But all of this was just a really preliminary analysis. And of course, anybody who's looked at this data knows that it's very noisy. So we didn't want to go ahead just with that, but we used a method we previously developed and applied to a yellow fever data set, and we're building a package now called Noisette to look at it. And basically the idea is to actually deal with noise in the data. So if you look at 2 replicates and you look at the frequencies, the counts in one replicate versus the other, you see there is a lot of noise. So we knew we needed to deal with that. So what we did is we calibrate a noise model using data from the same time point. And so what is the noise model? The noise model basically takes us from the frequency of real clones to the frequencies and the counts we see in the sequencing reads. So there's of course sampling noise, which we model as Poisson noise, but there's also noise that comes from differential mRNA expression bursting and cell loss and different things like that, so that we put into a long tail distribution such as negative binomial. So our noise model has 2 steps and takes us from the real frequencies that we cannot observe to the frequencies we actually observe in the data. So once we've calibrated the noise model, where this gives us a basis for probabilistic inference of the response. And so the second step is to compare two different time points and build an expansion model. In the expansion model, we assume that this expansion, but we also assume that there's a fraction of the clones which expand and a fraction which don't. And then we use the posterior probability to identify expanded clones. So we applied this to our data, comparing day 15 to day 85. And there we're able to see again the contracting, what we called previously the contracting clonotypes. This is day 15, so they contract in both donors now, which are color-coded, stably to all different time points. And similarly, if we compare day 15 to 37, we're able to see the the clonotypes, which we called expanded, although they actually expand and contract, they just have a peak at day 37, so the later response. And we see this large expansion in both CD4 and CD8 cells, the two donors again being very similar, with the fact the expanding one having a stronger CD4 bias, whereas the contracting ones being balanced between CD4 and CD8. And you also see here we added the pre-infection time points, and you see that the expansion is actually real compared to the frequencies that we saw at the pre-infection time points. So we also wondered what these clonotypes actually are. So we asked whether any of the responding clonotypes are found in VDJ-DB, in the VDJ-DB database that maps TCRs to known epitopes. And except for 3 minor exceptions, these clonotypes are responding clonotypes, so both contracting and expanding, do not map to any non-COVID specific VDJ-DB data. So they seem to be specific. If we look at them in more detail, I mentioned that we have a separation into memory and naive. I'm going to show you data for contracting, but the same holds for expanding. We see memory post-infection in both CD8 and CD4 for both donors. And if we take a closer look, we see that most of the memory is in the effector memory pool. However, we also notice a bit of long-lived stem cell-like memory in both CD4, CD8. We're doing the comparison here only for donor M because we only have the memory separation for donor M. But perhaps more interestingly is actually this information, which tells us that we see responding clonotypes at the pre-infection time point. So what I'm plotting here are are responding clonotypes, specifically the contracting ones, at different time points. So we see them in both donors, in both CD4, CD8, at the time points before infection. So it seems that there is pre-existing memory pre-existing memory of clones that we identify as COVID specific. And interestingly, if we again look in donor M, in CD4 cells, the responding clonotypes to the contracting ones at the pre-infection time point were mainly 6 in the central memory pool, and there was a phenotype switch to of these clones. This is not shown here in the data, but the clones here that are in central memory become effector memory post-infection. So there's a phenotype switch there. But as I said, the most important message is that there is both stable post-memory, but there is also pre-existing memory. So as I said, these are potential candidates for being COVID-specific clones, but I haven't shown you yet that they're COVID-specific. So this is what I'm going to do here. And to do that, I'm actually going to take advantage of the great resource that the community has built and the way we share data, and go to a study from Shumarodova et al., which is a tetramer study using this specific tetramer. And luckily, our donor M is also in their study. So donor M was sampled at day 5 post-infection and took part in that study. So we know the tetramer positive clones in donor M at day 25, and we can track them over time in our longitudinal data set. So we can see how often they appear at different sets in our data, and that's what this blue line is. So basically, we see that the tetramer frequency plus clones reproduce the contracting trend we identify independently from our longitudinal data. And what's color coded here are the frequencies of the actual contracting clonotypes that were independently identified in our computational study. So you see that a computational study identifies essentially all of the high frequency clonotypes that are found in the tetramer study. So this shows on one hand that our clones are COVID specific. On the other hand, it shows that this computational method is able to identify the large responding COVID specific clonotypes in a single patient without any cohort information. So, okay, so what do we learn about the actual clones? So let's look at motifs, because we know that responding clonotypes tend to be similar. So the first thing we verified this is true. We looked at the contracting data. We build links between similar sequences, so sequences that have the same V and J and differ by at most 2 amino acids. And we found that there are more links between these sequences than expected in a randomly sampled repertoire of a similar size. If we look at the class, so then once we have the edges, we build clusters. And if we look at the clusters are donor specific. That means donor M and donor W have different clusters. This is not surprising since we know they have the similar age A types. But if we take a closer look at the clusters, one thing that we're able to re-identify are clusters that correspond to the clonotypes that responded in the tetramer study, both in the alpha and the beta set. It's a fairly large fraction of the data set, but it is not the biggest cluster. So this is in donor M, and what can we learn about the different clusters? So here we turn to yet another data set from the community. This is the mirror data set that I think we'll hear more about. So this is an assay for doing epitope mapping. Basically, they took COVID specific, so this is from Adaptive. They took COVID specific antigen and tested them in a well plate against blood samples taken from people, and by doing combinatoric mapping, they were able to map specific epitopes to specific TCR, CD8 TCR beta sequences. So we take their data and we take all our responding clonotypes, we put them together, and again we build similarity networks of putting edges between sequences that have the same V and J and are less than 1 amino acid away in CDR3. And this allows us to identify clusters that we're able to link to 32 specific SARS-CoV-2 epitopes. And all of these clusters include clones that we identified as contracting. So now we have mapped our TCR clusters to SARS-CoV-2 epitopes thanks to the mirror study. And if we take a look at them, we know that this largest cluster so first of all, we reproduce the tetramer epitope and we reproduce its identity, but we find that the largest cluster, which takes up 21% of the CD8 response in donor M, corresponds to this specific epitope, and it's in fact an immunodominant epitope in donor M. So we can take another look at it and use another technique we developed for longitudinal alpha-beta pairing. So we take the longitudinal trajectories of alpha and beta clones and statistically map the change, their frequencies over time to figure out which alpha and which beta come from, receptor comes from the same cell. And if we do that, then it turns out that we can map the largest cluster in the CD8 TCR beta response to the largest cluster in the CD8 TCR alpha response. So we have a predicted pairing of this immunodominant response in donor M. And interestingly, the specific, the T cells that are specific to this epitope are also found in donor M in the pre-infection time point in the effector memory pool. So it seems that this is a clone that is cross-reactive, and it's also known that similar epitopes to this are known to be specific for non-COVID, so basic cold coronaviruses. So we do see cross-reactivity. And so that was all about CD4. Now let's move on to, sorry, that was all about CD8 because the mirror data and the tetramer was done with CD8. Now let's move on to CD4. So we can also identify clusters in the two donors in the alpha and beta pool chains, but unfortunately we do not have something data like mirror or tetramer data, well, mirror to compare to. So what we did instead, we went to another study. We went to a very large COVID cohort study that recently came out from Adaptive, and we compared that to a control study also from Adaptive, what's called commonly called the Emerson dataset. And now I'm gonna talk about donor W just for fairness, but the results also hold for similar results for, of course, different HLA types hold for donor M. And so we have, we're gonna focus on this large cluster identified in donor W, and we're gonna see to match it to large frequency clones in the COVID cohort and the control. And we're going to see that this the clones corresponding to this motif, the large cluster from donor W, are overrepresented in the COVID patients compared to the controls. So we asked the question, can we identify COVID-19 patients simply by the frequency of the TCR motifs? So what we did is we built a classifier and we compared the true positive rate versus the false positive rate in all donors for this specific motif, and unfortunately the answer is not really. However, we did not use any HLA information, so we asked ourselves, what if we use HLA information? Unfortunately, there's no HLA information in the COVID-19 cohort. So we turned to the control cohort that does have HLA information, and we used another idea developed by the community, this time by Will DeWitt and Phil Bradley and others, of HLA inference. So forgetting about COVID for a second, we just, since we know what the HLA type is in the control donors, we mapped large clones from the control donors to sequences in the COVID patients, and based on that, we inferred the HLA types of the COVID, of the people in the patients in the COVID cohort. And then we restricted our analysis to, so first of all, we could identify that this motif corresponds to this specific HLA type, which we happen to know is correct because we know the HLA type of donor W, and then restricted our analysis just to HLA matched donors, which are shown here in orange. And if we re-ask the question, can we identify COVID-19 patients by the frequency of the HLA motifs if we restrict to a given HLA type, the answer is yes. We get a much better classifier based on very small, on data from basically a small number of patients. And as I said, the same results hold for M. So in conclusion, I hope I've convinced you that we can identify COVID-19 specific response from longitudinal data, even if we only have one patient. We've shown this by validating our result comparing to other data sets produced by the community, for which we are very thankful. And we're able to show that by identifying motifs of COVID-specific response, this can be used for diagnostics and identifying patients that have undergone the infections. Biologically, we see two waves of a response, the fast expected one and then a second one, which is a little bit more mysterious. We see pre-existing COVID-19 memory, so this means that there is cross reactivity and that there is some sort of existing at least partial immunity, and then we see stable post-infection memory. And here is the data. There'll be an updated version of the manuscript available tomorrow morning on the archive with everything I mentioned, and the data is available here. So thank you.
Thank you, Aleksandra. That was very nice. So a few questions. One is you found the 3 hits in VDJ-DB. Can you remind me, was that looking at which time point and from which of the 2 patients?
So this is all data together.
Okay.
I don't remember which one is specifically. Basically, this is pulling everything together. These are the responding, so contracting and expanding TCR beta, everything we identified as a response. But it's important to say that this is the non-SARS-CoV specific VDJ-DB. There's also now COVID data in VDJ-DB, so that does not include it.
Okay, so one question that's come in on the chat is that the 3 seems to be high. Did you also do a comparison using randomly sampled clones to assess your hit number?
Sorry, 3 seems to be high?
Yes.
So 3 hits in VDJ-DB. Out of, I didn't tell you out of how much, how many, I guess. So if this is TCR beta and 2 donors, I mean, we're looking at around 1,000 clonotypes, because donor M TCR beta had about 600, over 600 responding ones. Donor W had less, or a bit under 300. Mm-hmm. And these are just the contractings, a bit smaller numbers for expanding. So, I mean, we did statistical tests for this. I mean, also the overlap is between Epstein-Barr virus. Mm-hmm. And the hits were HLA mismatched to the Epstein-Barr virus, I should add. So that also maybe makes them a little bit less exciting as hits.
Right. So is there any potential peptide overlap between those 2 viral genomes? For example, the EBV peptides that were the hits corresponding to the TCR hits you found. Is it possible those peptides are found in the SARS-CoV-2 genome?
Not really, no, I don't think so. I think, if you want to concentrate on overlap, what I would be more excited about is the overlap with the common cold coronaviruses for the immunodominant epitope of donor M. Mm-hmm. I think that's the exciting cross-specificity, cross-reactivity that we have.
Yes.
And there the epitope for the common cold differs by one amino acid from the epitope that's in mirror. So there's a Q that goes to an A, and so I think that's the more exciting one. This, I think, is
Yeah. No, agreed. So those hits that you were just referring to with the mirror data set, were those tracking back to the first peak at day 15 or the second peak at day 37, I think?
no, those are all 15.
Those were only pre-infection? Oh, all 15. Okay,
Yeah.
all 15.
They're all the ones that we call contracting. As I said, this maybe is a bit of a misleading nomenclature because they all contract in the end, but the ones that do not expand, that they basically that contract from day 15 onwards systematically, those are all the mirror hits, and the tetramer positive ones also.
And so that means there were no hits with the day 37 peaking clones?
Not in what we found, but
not what you found.
So what this means is we cannot conclude that the 37 peak is specifically COVID specific. It could be a bystander reaction. It can be maybe another infection that happened. It could be many other things. It could also be a secondary reaction just from, it could be B-cell induced. We do not see this in other data sets.
Okay. Interesting, yeah. So another question coming in is that your clones were identified because the infection was ongoing or very recent. How would you adapt your method, or do you think it would work sort of out of the box, applying it to patients who had experienced COVID-19, say 2 years ago?
So of course that's a hard question, and we're curious to have a look at that. So there are no patients that had it 2 years ago right now. We're also but we're going to do that. I mean, I think one thing we saw is that what is important is the sort of, as we probably all know by now, that the treatment of the samples, to get the samples sequenced as soon as possible, or at least prepared for sequencing as soon as possible so they don't sit around, that seems to have a big impact on whether we're able to detect something. Of course, there's a decay. We don't know what the decay timescale is. Judging on our data from, if we only had the 85-day time point, yes, then it would be much harder to say something, but maybe not impossible. So I think we'll have to look at that. Remember, these are also people with relatively mild symptoms. So one of the things we're trying to look at is people with different degrees of symptoms.
Yeah. So, and then I'll give last call to the attendees if anyone wants to post another question while I ask the current last question. Back to the mirror data, where all the hits you found were with the day 15 peaking clones, do you think that could be related to when the samples were collected for that mirror study? Were those maybe not far enough out that you could find hits with your later peak?
So the mirror study patients have quite a wide range of when they were sampled, but it could be. I mean, it could be. I mean, that's why I'm very careful to not say too much about this 0.37, because just because we don't see it doesn't mean that just because we don't see COVID's, among the clones we identify in that second peak, just because they don't come up in other studies, doesn't mean they're not COVID specific. It just means there's no study that has identified them yet. So it could be because of the different sampling. It could be because the MIRA study chose to focus on certain epitopes and not others. From I think they mostly focused on the spike protein. So I think we'll see. It's, as I said, it's a more mysterious peak, and we'll be curious to try and figure out what's going on there.
Yeah. And I guess, was it the later peak that was CD4 dominated?
Yeah.
And so I wonder, could those be, for example, regulatory T cells that are participating in ramping down, so that, and because these are patients who didn't progress to the sort of runaway inflammation?
Yes, exactly. So that was one of the ideas that came to us. Basically, this does seem to be some I don't want to say afterthought response, but I think that's a very good suggestion. So
excellent. Okay, well, yeah, this was a very interesting talk. Thank you so much for that. And any more questions for this speaker, you can reserve for the panel.